This is a presentation about enzymes by Sydney Bradshaw. So what is an enzyme? It is one of the three main types of catalysts. It's defined as a highly specific biological molecule that acts as a naturally occurring biological catalyst within cells. One important thing to note is that one type of enzyme cannot catalyze just any substrate. This is why there's so many different classes and types of enzymes. An enzyme is specifically made for a specific substrate. And you kind of relate this idea to a key fitting into a lock. One key cannot unlock just any lock. Some background information on catalysts is that catalysts are defined as substances that will increase the rate of a chemical reaction without being consumed by that reaction and without altering the overall equilibrium between the reactants and products. Because it is not consumed or degraded, it can be used for future chemical reactions. Some tips about enzymes: that they are usually large proteins with complex structures. They are not considered to be reactants, which is why they're not going to be shown in the overall stoichiometry of the reaction that is being catalyzed. And they will, however, they will be shown in the elementary steps. Most are temperature dependent and will not function at temperatures greater than 37 degrees Celsius. What is the role of an enzyme? And this will be described in a series of simplified steps. First, that substrate is going to bind to an enzyme and form what is called an enzyme substrate complex. More specifically, they will bind to an area called an active site on the enzyme using intermolecular interactions or bonds, and this might include hydrogen, dispersion, or covalent bonds. Once that substrate binds to the active site, it's going to force that substrate into a very specific orientation, which is just going to stress and weaken the bond. And just also to note, a chemical bond is a form of stored energy or potential energy, and a certain amount of energy is going to be required to break that bond. So when we say that we're weakening the bond, we're saying that not as much energy is going to be required to break the bond between the molecules. And this is how an enzyme is able to effectively lower the activation energy of a reaction. And activation energy is pretty much just an energy barrier or a requirement of how much energy is going to be required in order to complete the conversion of reactants to products. So when we say that we lower the activation energy, this means that the reaction is going to take place at a faster rate towards equilibrium because not as much energy is required to fuel it. And this does depend on temperature. So once those products are made, they're going to be released from the active site. So here's a diagram just kind of showing the process that we just described. You're going to see this substrate in green and the enzyme in yellow. So the substrate is going to bind within the active site of the enzyme, and you can kind of see that these two have a very specific shape to themselves so that they can only form with this part of the enzyme. So once it binds to it, the bonds between the substrate, within the substrate, will be effectively weakened, and they will convert to products. And once they are formed into the products, they will be released from that active site. And here is it's an energy diagram. It's going to pretty much show the difference in the energy requirements for a reaction to occur when a catalyst is present and when it is not. So in the green, we're going to see this is when a catalyst is not present. So you can kind of see the amount of energy that is required to reach that complex is much higher than the line depicted in purple. And the one in purple, you can kind of see by this label, is that this is the catalyzed reaction. So this is when an enzyme is present. You're only going to need this much energy to pretty much convert those reactants to products. So you can see how this is this is more favorable energy-wise to occur within the cell versus one without a catalyst. And so my example is going to be sucrase, and it is a hydrolytic enzyme. It's a hydrolase that will add water molecules to a compound to cleave those bonds. So it's known for catalyzing the hydrolysis of sugar or sucrose to release two products, glucose and fructose. And you kind of see in this not energy diagram, but this diagram, sucrose enters, it bonds with the active site, and this is where a water molecule will be added, and it shows once it's added that sucrose is going to break up into glucose and fructose. Lastly, this is another diagram that shows the enzyme catalyzing the actual reaction. So I like this diagram because you can actually see the bond between the sucrose molecule, how this is going to be weakened, so that once this breaks apart, you're going to have two separate products formed, glucose and fructose. And this enzyme is commonly found within the intestines, and it just breaks down that sucrose into smaller subunits so that they can be used as forms of energy for the body. And this concludes my presentation. Thank you for listening.